morning. Before I begin, I would like to thank the organisers for this wonderful opportunity. Thank you. So I'd like to introduce myself. My name is Kirsty Lewis, and I'm currently a research assistant within Professor Nikolai Zhelev's research group at Abertay University in Dundee. And unfortunately, Professor Zhelev couldn't be with us today due to family reasons. However, he's asked me to come along today and present some of his research to you all. And he has been investigating novel stem cell-based approaches in the development of cyclin-dependent kinase inhibitors as new therapeutics in the field of cardiology. So I'll start off with just some background details and then I'll get into what we've been doing. So cardiovascular diseases is an umbrella term for all diseases of the cardiac muscle and circulatory system, including coronary heart disease, stroke, and other cardiovascular diseases such as cardiomyopathies, all of which can proceed to congestive heart failure, whereby myocardial performance is insufficient to meet the metabolic demand of the vital organs and tissues. It remains one of the leading causes of morbidity and mortality worldwide, accounting for 17.3 million global deaths per annum, a number which is expected to grow even higher to about 23.6 million by 2030. So this is a huge health issue worldwide. The basis of our research has been focusing on hypertrophic cardiomyopathy. Now, this is a common form of CVD, which is estimated to affect 1 in 500 individuals in the UK alone. And the condition is characterised by this abnormal enlargement of the ventricles of the heart. And this is as a result of increased cardiomyocyte cell volume. This then leads to thickening and stiffening of the ventricles of the heart, particularly the left ventricle, which can then prevent the chamber from filling properly and leading to numerous abnormalities, including interruption of the blood supply. In severe cases, it can lead to sudden cardiac arrest. The disease is pathological and physiological and is common in individuals who undergo chronic exercise. It's actually reported to cause 36% of CVD-related deaths in competitive athletes. So as with many other forms of CVD, a curative pharmacological treatment is yet to be established for HCM. Current treatments focus on improving patient quality of life, and this is through controlling symptoms such as elevated blood pressure, and they don't directly target the disease process. However, our research is trying to attempt to resolve this medical issue, and we have been doing this through studying the CDK9 pathway. So involvement of the cyclin-dependent kinase pathway has been identified as a fundamental factor in the induction of hypertrophic cardiomyopathy. CDK9 is broadly distributed within all tissues throughout the body and is especially highly expressed within terminally differentiated cells. It performs essential roles in several biological processes, including cell growth, protection from apoptosis, and also cell differentiation. However, this molecular mechanism is hyperactivated by hypertrophic stimuli, such as increased biomechanical stress caused by chronic exercise. So that's why the disease is common in athletes. And this hyperactivation leads to increased phosphorylation. And this is at the C-terminal domain of RNA pol2. This in turn causes a promotion of RNA elongation and therefore increases protein synthesis, which increases cellular volume and causes the ventricles of the heart to enlarge. So this can obviously have detrimental effects on the blood circulation. So we have suggested that by using small CDK9 inhibitor molecules, which target the inactivation of the CDK9 pathway via inhibition of the phosphorylation, They could be used to prevent the onset of this hypertrophic enlargement in cardiomyocyte cells. So looking at drug development, now in order to screen candidate drugs for therapeutic interventions, such as these CDK9 inhibiting compounds, a reliable test model is required. However, currently the lack of physiologically relevant preclinical models, as well as the obvious difficulties in obtaining cardiomyocytes out with the human body, and obviously their inability to reproduce in vitro. These have both been identified as the underlying cause of inefficacy, where preclinical trials have often then looked to animal models. But as I'm sure you're all aware, there are numerous interspecies differences between animals and man, and therefore animal models fail to fully recapitulate the human physiology, and thus they don't faithfully replicate the human cells and don't predict human cardiotoxicity. So this has pressed the pharmaceutical industry to explore alternative methods for screening novel CVD drugs. So human embryonic stem cells hold great promise in bringing new and effective cardiovascular treatments to the market. And they do this through providing a testing platform for preclinical drug screening. The cells are derived from human blastocyst stage embryos, which are surplus to the requirements of in vitro fertilization. And they do have the capacity to self-renew indefinitely, and they provide a readily available source of all the cell types of the human body, which is a considerable benefit over the use of animal cell lines. Stem cells can be continuously propagated out with the body in vitro and stimulated to undergo directed differentiation into de novo cardiomyocytes, and they represent all phenotypes of the human body. Cardiomyocytes derived in this manner aim to overcome this lack of physiologically relevant preclinical models for toxicology testing, and they do this by providing a novel system that's scalable, reproducible, and from an inexhaustible source. So here we see the process whereby these embryonic stem cells are derived from the human blastocyst cells. So they undergo differentiation via various growth factors, and that's into the cardiomyocytes. And at the bottom there, we also have induced pluripotent stem cells, which are derived from fibroblasts taken from tissue biopsies. And they can be undergo reprogram back to the embryonic stage, or they can also be directly differentiated into cardiomyocytes. So we've been looking at the embryonic derived stem cells. So this is our model. Our research group have established this model, which is a cell-based assay where the human embryonic stem cells undergo differentiation into cardiomyocytes, shown here on the left. They're then clustered to form mini hearts, which are essentially spheroids, and that's shown on the right there. And hopefully this video works. Cool. So these mini hearts are in fact capable of spontaneous rhythmic contraction and beat just like a human heart. They also generate action potentials and ionic currents characteristic of cardiac cells. They beat at approximately 30 beats per minute, and these particular mini organs are 1 millimetre in diameter, and they consist of approximately 40,000 individual cardiomyocytes. So the mini hearts can be cultivated within media in a 96 well plate, and this provides the opportunity to induce diseases within these mini hearts, for example, cardiac hypertrophy. And this creates a diseased phenotype, which is ideal for medium throughput drug screening of novel therapeutic agents. and obviously avoids this contentious use of animal models. For our investigation, cardiac hypertrophy was induced using the hormones angiotensin II and endothelin 1. In our hands, angiotensin II appeared to work slightly more effectively, and an approximate volume increase of about 29% was seen in the hypertrophic mini hearts. So now that we've got our test model, we can now screen novel compounds. And we have been looking at cyclin-202, which is a novel CDK inhibiting compound developed by the pharmaceutical company Cyclin in Dundee, Scotland. And the compound is known to inhibit CDK9 and has already been tested in patient clinical trials and shown to have positive effects as an anti-cancer drug candidate. It's actually been tolerated well by the patients. The drug works by binding and blocking the ATP binding pocket of CDK9, and this prevents phosphorylation of the C-terminal domain of RNA pol II. So in order to test this innovative discovery, numerous methods were carried out, including assessments of cyclin-202's stability and cytotoxicity, and then went on to do our drug dose response assay involving the mini hearts, and obviously data analysis. So the HPLC results demonstrated that cyclin-202 is extremely stable over time. And a standard MTT assay showed an IC50 of 10.48 micromolar. So going on to the drug response assay. As you can see, following this, the mini heart is treated with Ang2 to induce hypertrophy. That's in the middle figure there. They show a visible increase in mass compared to the control. And the hearts co-treated simultaneously with Ang2 and the drug show no visible increase in mass, and that's in figure C. So this shows that the onset of the disease is prevented by the drug's presence. So here we have an Excel graph, and whilst it confirms that the control hearts display no percentage volume change, and that Ang2 and ET1 cause a significant mean percentage volume change of between 23 and 29%, confirming that cardiac hypertrophy is mimicked within these mini hearts. The compound on its own has no statistically significant effects on the cardiomyocytes, that's shown in the pink, and co-treatment of the hormones alongside various concentrations of the drug prevents the onset of the disease. They're shown along the end towards the right there. And to confirm that the drug is in fact targeting CDK9, a Western blot was performed, and phosphorylation of the C-terminal domain of RNA pol II is increased during Ang2 and ET1 treatment. You can see that in the middle. However, this phosphorylation is then decreased by the addition of cyclin-202, indicating that the substrate is inhibited by the drug in vitro in the cardiomyocytes. And here we can see that anti-troponin T labels the cardiomyocyte-specific protein troponin T red, and that's in the first column to the left there. Cardiomyocytes treated with anti-SER5 primary antibody are stained green, and the intensity of the staining corresponds to the degree of phosphorylation. As you can see, phosphorylation levels are moderate in untreated cells and increase during Ang2 treatment in the middle box there. By treating the cells with cyclin-202 alongside the hypertrophic stimulus, phosphorylation is limited due to CDK9 inhibition. And on the right, we have a merged column there. So our conclusions drawn showed that cardiac hypertrophy can be successfully modelled in human embryonic stem cell derived cardiomyocytes. And hypertrophy can be induced using hormones and prevented through the co-treatment of the hormone alongside this Cyc202 compound. The compound was found to be suitable for further clinical development of drugs for treating this serious disease. And overall, this mini heart organ model aims to overcome the current lack of physiologically relevant preclinical models. And it does provide a novel system that is scalable, reproducible, and from an inexhaustible source. Moving on from here, some further research. This will involve the synthesis of analogues of the compound, and each will be tested for their selectivity, stability, potency, and bioavailability. From there, the best analogues will then be put into preclinical studies, and then hopefully progress on to clinical trials. As I'm sure you're all aware, the drug discovery process is extremely lengthy. However, this compound is at an advantage because it is already being used for patient clinical trials as an anti-cancer drug. So we already know how it works within the human body against normal, healthy cells. So hopefully this compound and novel heart assay will bring us even closer to finding a curative pharmacological treatment for the disease, especially for those at high risk of developing the condition, such as athletes. And finally, I would like to say a big thank you to Pharmaceuticals for kindly sharing their drug through a material transfer agreement and for their contributions to this project. And I would like to thank you all for listening today. I'd be happy to try and answer any questions you have regarding Professor Zhelev's work. I've also put in his email address there. Please feel free to email him with any questions. He's very open to any communications. He's also very open to any collaborations if anybody's particularly interested in this mini heart organ model. So thank you very much.
Thank you very much for this interesting presentation. We have plenty of time for questions, 10 minutes. Or comments, feel free to ask and share. Yes? Yes?
So do you use human embryonic...
Can you just introduce yourself?
Oh, sorry. My name is Michael Casper. So did you use human embryonic stem cells or IPS?
It's human embryonic stem cells.
And is there a reason for one other than the other, or do you only use one kind of...
We have used both. And they both show similar results. It's just in this one, I've just chosen to show the embryonic side instead of using both. I've just chose embryonic stem cells. But we have tried both, and they both show similar results.
Yes.
I was wondering, like for the CDK, I forgot to introduce myself. Tulani from University of Guelph. I was wondering for this CDK9, the
Kinase.
in addition to the pathological effect that you have mentioned on the heart, is there an important physiological role?
I've just been looking at in terms of the physiological side for sort of athletes, endurance training, when they undergo a lot of exercise, it obviously puts strain on the heart and causes hypertrophy. Because hypertrophy is obviously a normal process that's involved in, from childhood to adult. Hypertrophy is a normal, healthy process. It's just it is hyperactivated in athletes. So that's where I've been focusing. So I'm not entirely sure on sort of pathological side. It's not really what I've been looking at. So
I see. I was just wondering because maybe CDK9 maybe it's not just being expressed in the heart; they're expressed in other tissues.
Yeah, it is expressed in all other tissues. Yeah, it's just that it's hyperactivated during this hypertrophic stimulus.
Right. So my question is, if they play other physiological role, what about the long-term side effect if you need to use this inhibitor for a long period of time?
I know. Yeah, that is something that's needing further research, obviously. Right now, as I said, patients are showing to tolerate this drug very well, but it is obviously in early stages right now, so we would need to do further testing. Yeah.
More questions? Okay, go ahead.
In your future study, you propose to identify analog for CYC202. If this one is working well based on what you are talking about, why would you like to identify a new one, which involve another linking process?
Yeah, I think we're looking at developing analogues because the one we've been using right now is very stable. It's like extremely stable.
Right.
So I think we'd need to do some sort of chemistry work on the drug to sort of bring that stability down, because it can't be, obviously, if you're giving that to someone, you can't have that drug within someone's system for that length of time. So that's why I think we're developing analogues, just to sort of tweak little things like that. Yeah.
Yes. What is your name?
My name is Ali, student at Aston University.
Okay.
Is it going to be used as a prophylactic or for the treatment?
For preventing the disease. What I've been showing you just now is looking at preventing the onset of the disease, so taking it as a preventative treatment. However, we have looked at reversing the disease process, which is shown to have positive signs. I haven't shown you that today because I wasn't involved in looking at that side of the experiment, but I know that it is currently under investigation, and it is shown to sort of reverse the hypertrophic state. Yeah.
So, if there are no more questions, on behalf of the organizing committee and Omics International and the editors of Journal of Biotechnology and Biomaterials, Journal of Bioprocessing and Biotechniques, and Journal of Microbial Biochemical Technology, I would like to present to you and to your co-author this certificate of recognition for your valuable contributions and worthy keynote presentation.
Thank you. Thank you very much.
Thank you very much.